Good morning, it's me Afsal. In this video, I'm going to discuss about enzymes. In this module, I will be discussing the following: enzyme, enzyme catalyzed reactions, chemical nature of enzymes, characteristics of enzyme and enzyme catalysis. In the beginning, we have to briefly define enzymes. Then, what are enzymes? Enzymes are biological catalysts which catalyze the various biochemical reactions in the cells. They are capable of acting independently of the cells. Majority of enzymes are proteins with catalytic capabilities crucial to perform different processes. Metabolic processes and other chemical reactions in the cell are carried out by a set of enzymes that are necessary to sustain life. The initial stage of metabolic process depends upon the enzymes which react with a molecule and is called the substrate. Enzymes converts the substrate into other distinct molecules and are called the products. Then enzymes are of two types: endoenzymes and exoenzymes. Then first one, endoenzymes. Enzymes which function inside the same cells which produce them are called endoenzymes or intracellular enzymes. Respiratory enzymes are some of the examples for endoenzymes. The next one is exoenzymes. Enzymes which function outside the cells which produce them are called exoenzymes or extracellular enzymes. Digestive enzymes like amylase, pepsin, trypsin, etc. are some of the examples for exoenzymes. Next, we have to discuss enzyme catalyzed reactions on the following slides. We have to deal with some typical examples of enzyme catalysis. At first one, we have inversion of cane sugar. Here, the enzyme invertase catalyze the hydrolysis of sucrose into glucose and fructose. Then next we have conversion of starch into maltose. In this reaction, the enzyme diastase catalyzes the hydrolysis of starch into maltose. Next, we have conversion of maltose into glucose. Here, the enzyme maltase catalyzes the hydrolysis of maltose into glucose. Next is the decomposition of urea into ammonia and carbon dioxide. In this reaction, the enzyme urease catalyzes the decomposition of urea into ammonia and carbon dioxide. Next is the conversion of proteins into peptides. Here in the stomach, the enzyme pepsin, which is secreted by the chief cells of the stomach. Pepsin catalyzes the conversion of proteins into peptides. Then in the intestine, the enzyme trypsin, which is secreted by pancreatic cells, catalyzes the hydrolysis of proteins into amino acids. Here are some of the examples of enzymes and the reaction they catalyze. First one, lactase. Lactase converts lactose into glucose and galactose. Amylase converts starch into glucose. Pepsin converts proteins into amino acids. The DNA polymerase and RNA polymerase converts deoxyribonucleotide triphosphates and ribonucleotide triphosphates into DNA and RNA respectively. Next, we have to look the chemical nature of enzymes. Chemically, enzymes are globular proteins of high molecular masses. They may be simple protein enzymes or conjugated protein enzymes. Simple protein enzymes are those whose molecular structures are made up of alpha amino acid units only. Pepsin, trypsin, urease, amylase, etc. are some of the examples for simple protein enzymes. The next, conjugated protein enzymes. Conjugated protein enzymes, otherwise called complex protein enzymes. They contain a non-protein portion attached to the protein portion. The protein portion is called apoenzyme, while its non-protein portion is called cofactor. The apoenzyme cofactor complex is called holoenzyme. The next is the cofactor. Cofactors are indispensable for the catalytic activity of conjugated enzymes. That is, neither the apoenzyme nor the cofactor can be active by themselves without the presence of the other. Only their combination, that is, holoenzyme, can show catalytic activity. Cofactors confer activity on enzymes by acting as a bridge between the enzyme and the substrate or by taking part in the reaction. Then the cofactors are of three types: prosthetic group, activator, and coenzyme. First one, prosthetic group. If the cofactor is firmly bound to the apoenzyme, it is called the prosthetic group. The iron porphyrin complex of cytochromes, heme in hemoglobin are some of the examples for prosthetic group. The next comes the activator. Activators are inorganic metal ions. They form coordination bonds with side chains and also form one or more coordination bonds with the substrate. Then magnesium, calcium, zinc, manganese ions are some of the examples for activators. Then, for example, the alcohol dehydrogenase is activated by the zinc ion, and the enzyme arginase is activated by manganese or cobalt. The next is the coenzyme. Coenzymes are organic compounds like a B vitamin. Their association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as a cofactors in a number of different enzyme catalyzed reactions. For example, thiamine pyrophosphate, pyridoxal phosphate, etc. The next, we have to discuss the characteristics of enzymes and enzyme catalysis. Here, we have to discuss the functional characteristics and the condition characteristics, that is, the factors influencing enzyme action. So, first, we have to look functional characteristics. The first point, they are high catalytic efficiency of enzymes. Enzymes are highly efficient catalysts. They are a specialized class of proteins which can increase the speed of biochemical reactions even up to 10 million times that of uncatalyzed reactions. The next point is the need for very small quantities. Even extremely small quantities of enzymes can speed up the concerned reactions to a very great extent. Even a single enzyme molecule may be capable of bringing about transformations in a million substrate molecules per minute. The next point, the unalterability of enzymes. Enzymes are not broken down or changed in the process of their catalytic functions. This means that an enzyme is as potent at the end of a reaction as its beginning. Then fourth point is the specificity of enzymes. The enzymes are highly specific in their catalytic action. That is, a particular enzyme generally can catalyze only a particular reaction or a particular type of reactions. This is the specificity of enzyme action. For example, the enzyme maltase can catalyze the hydrolysis of maltose but not that of sucrose. Likewise, the enzyme urease can catalyze the hydrolysis of urea only. So, the next point is the ability to maintain the position of equilibrium. Enzymes speed up the attainment of equilibrium in biochemical reactions. They do not, however, alter the position of equilibrium in any manner. These are everything about the functional characteristics. Then we have to move on to the condition characteristics or the factors influencing enzyme action. First is the temperature dependence of action. Enzyme activity is sensitive to temperature changes. For each enzyme, there is a characteristic temperature at which it shows maximum catalytic activity. It is called optimum temperature. This means that the rate of the reaction that an enzyme catalyzes becomes maximum at the optimum temperature. Most enzymes in the human body have optimum temperatures around 37 degrees Celsius. For plant enzymes, optimum temperatures generally lie in the higher range of 40 to 60 degrees Celsius. Enzymes are denatured by heat and hence their activity is destroyed at high temperatures. The next is the pH dependence of action. Enzyme activity is pH dependent. Each enzyme shows maximum catalytic activity at a characteristic pH. It is called the optimum pH. For most human enzymes, optimum pH lies around 7. However, gastric enzymes act under very acidic conditions, that's around 2. Next is the activation by the presence of cofactors. The catalytic activity of most enzymes is enhanced by the attachment of a non-protein part called a cofactor to their protein portion. Cofactors confer activity on enzymes by acting as a bridge between the enzyme and the substrate or by taking part in the reaction. The next is the deactivation by the presence of inhibitors. Catalytic activity of an enzyme is decreased or completely destroyed by the presence of certain substances. They are called inhibitors. An inhibitor functions by interacting with the active functional group, the active site or active center of enzyme, and thereby inhibiting the function of the enzyme. Heavy metal ions like silver, mercury, lead, etc. inhibit the activity of the enzyme urease. Likewise, the activity of the enzyme succinic acid dehydrogenase is completely inhibited by the presence of malonic acid. Enzyme activators and inhibitors are collectively called enzyme regulators or enzyme modifiers. These all are everything about the enzymes, and my topic is over here now. Thank you for watching.